So let us talk about a certain group. They could be chemicals or organisms which are derived from natural products, or these are actually natural products or natural pesticides. So let us look at the natural products or biopesticides and their benefits. Now, biopesticides are pesticides derived from natural materials such as animals, plants, bacteria, viruses, and certain minerals, unlike our typical pesticides. But they more or less do the same function. They are used to control pests such as insects, weeds, and fungi. So here I've got a schematic, or I've got something which is trying to demonstrate what biopesticides can do. They can be derived from, they could be plant extracts which can kill an insect, or it could be bacteria or fungi which can actually kill the insect, but it's not going to harm the crop. Now, what are the target pests? Insect pests, diseases, fungi, bacteria, and viruses, or even weeds. Now let us look at the classification of pesticides or of biopesticides. We have microbial pesticides, biochemical pesticides, or plant incorporated protectants known as PIPs. So there are three categories: microbial, biochemical, and plant incorporated protectants, PIPs. What are the microbial pesticides? These are made from microorganisms such as bacteria, fungi, and viruses. Microbial pesticides can kill pests directly or by producing toxins that are harmful to the pests. One of the examples is bacterial thangyriensis, or BT, which can control pests. Biochemical pesticides, these are naturally occurring substances that can control pests. Some biochemical pesticides interfere with the mating or feeding behavior of insect pests. Now let us look at the plant incorporated protectants, PIPs. These are proteins or other molecules that are produced by plants that have been genetically modified to contain genes from other organisms. PIPs can kill pests directly or by making the plants resistant to pests. So we have got fungal substances that plants produce from genetic material that are added to the plant, e.g. Bt in cotton. So Bt gene was taken from the microorganism and inserted in the cotton plant so that the cotton plant can produce toxins which can then kill the insect pests. So we have a transgenic plants which produce biodegradable protein with no harmful effects to animals. That is the good thing about Bt gene. So here is a scheme where we have the Bacillus thuringiensis with that Bt gene on the DNA, which is inserted on the maize, or it could be cotton. Now, when that maize plant or cotton plant has that gene, if the borer tries to eat the maize, it's going to get the toxin. Then of course it will die. So we have made maize toxic or cotton toxic to the pest. Now, what are the major sources of biopesticides? Tephrosia vogeri, it's widely used as a pesticide plant, and it can also improve soil fertility. It contains rotenone, which does the killing of the pest. Tithonia diversifolia, this is a weed with insecticidal properties. It contains sesquiterpene lactone. We have got Nicotiana tabacum. Even tobacco itself contains Nicotiana alkaloids, which can kill whitefly, thrips, and even bollworm. Bacillus thuringiensis, this is a pathogen of the American bollworm, so it can kill the bollworm. So Bacillus thuringiensis can be a source or can be used as a biopesticide to kill American bollworm. We've got Trichoderma species, which are effective against soil-borne diseases such as rots. Could be used for dry-land crops such as groundnuts. They stimulate host plant defenses as part of the mechanism. Now, if we look at the plant can have a lot of these substances, which we call allelochemicals. Some have herbicidal effects, some act as insecticides or fungicides, and indeed some are poisons. Some could be pharmaceutical products. So we can exploit this route to control our weeds or fungi or insect pests. These are some of the allelochemicals which are produced by plants with a potential to have either herbicidal or insecticidal characteristics. Here I've got a table with a source of allelochemicals maybe you'll look at it closely if you want to see more examples where biopesticides could be, derived from. Here I've got the bottle brush tree, Callistinon citrinus, which is common in Australia. We also have it in this country, but I don't think it's the same type. So if we look at the discovery of mesotrione herbicide in 1977, the allelopathic weed control near bottle brush tree, Callistinon citrinus, led to the identification of leptospermon, a natural product. This led to the development of sarcotrione and mesotrione. And Zeneca Agrochemicals developed mesotrione for use in maize. The herbicide is now sold as Galago for pre and post-emergence applications in maize. So this was now fine-tuned to get that herbicide originating from the plant. So what are the mode of action of these biopesticides? These could be metabolic poison, growth regulators, gut disruptors, neuromuscular toxins, non-specific inhibitors, antagonism, predation, parasitism, and antibiotics. Now, what are the advantages of using biopesticides? They are often less toxic to humans and the environment. They can be more targeted in their activity. This means that biopesticides are less likely to harm beneficial insects such as pollinators. So they have a potential to be used in integrated pest management. These are the things which could be ideal. They often break down more quickly than synthetic pesticides, which reduce the risk of pollution. So these are the things which we need. No resistance problems. Probably you may be interested to note some of the research which we do, focusing on these. Maybe we may end up with the biopesticides. On weed control, we have been evaluating the allelopathic effects of Lantana camara, green manure cover crops, sorghum, to see their effects on weeds. Indeed, we have done a quantification of sorghum, which looks like atrazine actually in terms of its activity, from a number of sorghum accessions from 8 Southern African countries. We collected a lot of germplasm. Here we're working with a PhD student, Professor Tibugari, who is now at NAST University. He did a PhD on these things here in this department. Probably before I move forward, you may also be interested to note that Dr. Rugare did a DPhil on green manure cover crops, identifying aerochemicals, which could have a potential to be used as bioherbicides. Then plant pathogens, we've got the evaluation of plant extracts on Phytophthora infestans and Alternaria solani. We have Dr. Ngadze doing this kind of work. Use of moringa, oreifera, eucalyptus, lantana camara, garlic, papaya, neem. It's a potential area to produce biopesticides. Actually, there was a student, Choga, who was at the point of producing a biopesticide. He was in the hub for quite some time, trying to develop this product. Then plant pathogens again. We have a streptomyces, trichoderma, effects on fusarium. Stored products. We have Professor Mvumi, who has done work on Eucalyptus nigra, Lipia javanica, to find the effects on weevils. Then we have got also evaluation of Lipia javanica, Zumbani efficacy on ticks. I think this was done by livestock research at the animal science or livestock department. So in conclusion, we can say that there is a high potential to develop biopesticides to target weeds, insect pests, and plant pathogens in this country. But what is needed is collaboration among scientists from different disciplines, and this is urgent. Once there is a good collaboration, I'm sure the products could come out with a potential to be used. So I will stop here.